My name is Marina Panova. I am a researcher in Gothenburg University in Sweden, and right now I'm sitting at Tjärnö Marine Biology Station. We want to develop good tools to be used in biodiversity monitoring, because there is a great potential in environmental DNA to replace our traditional monitoring methods when we either catch the organism or take the samples, and eDNA samples are completely non-invasive with just a sample of water. It also can be potentially more time and cost effective, and also it doesn't require in-depth taxonomic expertise. You don't have to be an expert in all different group of organisms to determine the species. You compare DNA to the reference databases with DNA for different species. So we think that it could be a really useful and great tool to analyze biodiversity in the future, already now. More time to do the research and more focused time to do the research when you also have all the other duties that we do in academia. Also, would be nice with a large finance project. That's what required to have a solid research program. When you can try and get the complete picture, not only answering disparate questions, important questions, but you would need more time to study the field, because environmental DNA is quite new field. We know very little how DNA behaves in the environment, how it's released, how long does it stay in the water, for example, what happens afterwards, and is there differences between different organisms and so on. So there are so many questions you need to study before we know we can use those methods reliably. When we know when it works, when it doesn't, if it doesn't, why. To use the methods in management and conservation, they have to be reliable. We have to be sure, or at least know what's the certainty of our results. If we find this species, or if we don't find this species, how certain we are. And this field requires quite a bit of research before we can actually take the tools and give it to the management. I'm in the Department of Marine Sciences, a relatively new department of our university, and it's basically combined different nature science discipline. It's geology, chemistry, oceanography, and biology. So it's very interesting for me as a biologist. Every time I listen to my colleague, I learn something new. How you can tell the age of the sediment, how the current carry around the marine organism, what happens with organic matter in the ocean, and what the role of different organisms. All the things that for me is working with genetic and molecular biologists quite new, so I'm learning a lot. I'm just here at the Tjärnö laboratory. We have a really great molecular laboratory lab, and we have a great group of researchers that are using DNA, genetic tools to study marine organism, evolution of marine organism, adaptation in marine organism. So it's really inspiring, and there's many people that you can ask questions, ask for help, and so it's a very great place to work. From the beginning, I was, as many people, I was so fascinated by the diversity of the nature, all the different plants and animals. My best thing to do in the weekend was to go to the zoological museum and look at the collection when I was at school. So that motivated me to go to the university and to study biology. So I have my exam in invertebrate zoology, all different group of invertebrates. And then when I was studying biology, I was very fascinated by molecular biology, about DNA and proteins and building stone for all this great biodiversity that we see. And there are small changes in DNA and proteins that can make big changes in the organism. So that's why in the end of my undergraduate study and for my PhD, I moved to molecular biology side. in many ways, yes. When I was doing my undergraduate and graduate studies, I met many excellent scientists, like, people that were really burning for the science, driven by passion, curiosities. Now I know that actually most of the scientists are like this. They were not an exception. And in some way, actually, it turned out for me even better than I could imagine, because rapid development of the field. When I was studying molecular biology and genetics as an undergraduate, it was all about few model organisms. None of them were marine. And even when I started my PhD, there was very little genetic information in databases on marine organisms. And there was very few tools that you could use to study genetics of marine organisms. And then we have this rapid development revolution in DNA sequencing methods. So now we can study the genome of any organism we like. We can analyze this environmental DNA, which are very, very small amount of the DNA left by organism. So when I was studying at the university, I would never imagine that I would be able to do all of this. There will be such a freedom to choose what you want to study with DNA. I think DNA method in general, to find something new. It doesn't have to be big. For example, with environmental DNA, we develop a method to detect target species. It could be an invasive species, it could be an endangered species. So for each species, you have to develop the methods based on its DNA sequences, and to develop one, to test it and see that it works, and to pass it to people who want to use it. That I think it gives me really satisfaction. Or find an explanation to some variations that we see in the DNA sequences, and to understand why it's like this. Things like that. Find something new. It can be a really small thing. Solving problems, when something doesn't work, when you test new protocol and it doesn't work, you don't get DNA out of your samples, or maybe you develop a method to detect new species, you test it and it doesn't work. So then you go home and you continue thinking why it is, what should I change, why it's like this, and then you have a plan what to do tomorrow. If you think about working in academia, the one thing that many other people dislike is unsecure funding. When you rely on external projects that are short, and you never know if you will be able to continue in some years, and also maybe exaggerated competition for funds and position. I understand that we have to be effective how we use research funds and choose the best possible project. But I think when we are in the situation when we have a success rate in research called about 10-15%, and we spend more and more money writing the proposal and then evaluating the proposal, so instead of doing research, I think it's probably not the best way to use the time of researchers. And of course, it's also very frustrating that we still hear about examples of inequality in academia based on gender or national background and so on. I think we could do better in academia. So this is things that I don't like. I definitely don't like it's a cleaning freezes from old samples, for example. That is something that I have to do. I have samples from many years. That's I don't like very much, but otherwise I actually like all of it. Simple protocol, the ones that you've done many times, that they're gonna work and you get your results. But probably the most exciting is if you try something new. If you do it for the first time, you don't know what you get. And I don't mind if it doesn't work, because then you have to do the research, there's some thinking, and get it work. So that's probably the most interesting part. When you have to do, for example, lab work or analysis in a hurry, when you don't have enough of time. Then I enjoy doing both lab work and analysis, but when you don't have enough time, I don't like them. And then, it's unbelievable what silly mistake you can make when you are in a hurry. A week later, you look at it, how can I do it? So yeah, that's I don't I don't like it all. Well, I like PCRs because that's the whole all testing is we do the PCR and then we look at the result on a gel, for example, or sequence the result. Did it work or not? That is that's very great. I remember how I was fascinated the first time we did PCR. You know, that you mix these solutions like DNA solutions, they are completely transparent. You put them in the machine, you put some temperature, you don't see anything. And then all of a sudden when you do the gel and stain, and you have the bands, and they're exactly of the size that you expected. And then you put them in the DNA sequencer, and they read the sequence. And this is the sequence what you expected. That is now I also do a lot of bioinformatic analysis, and so I do my DNA samples preparation, and then we sequence them, and then I analyze the results. And it's also, I like it very much when we do the whole procedure, and sometimes you see what happens in the lab, it affects the result. So you can make connections. So I know how it looks in the lab, and then I see how the results look like. And every time I analyze new samples, I learn something new. So I can predict from how it looks in the lab, maybe what I get in the results. Oh, that's a difficult question for a researcher. We've been talking now about the insecurity, and I'm not a faculty member, I have a research position, so it's difficult to imagine the future. I mean, I've been feeling insecure and worried many times, but each time it's turned out to be good. There was new opportunity to do research work and even to do the research I want to do. And so I hope it will continue this way, that I can continue work in academia or maybe in a research institute. And I would very much like to continue to work with environmental DNA because there is so much potential, but also there are so many questions to answer. And also there is such an interest and demand actually to have the methods and protocols that work and tested from environmental agency, from municipalities, other authorities. So I really see that the research that I do, it can be useful in a very short run. We are two working places is because we are located about 160 kilometers from Gothenburg, so I'm working in both. This is a station belonging to Gothenburg University. My project is about environmental DNA, how we can use it in biodiversity monitoring. So environmental DNA is the DNA traces that all the organisms live in the environment. It comes with the skin, hair, mucus. So if you take a sample of water or soil or sediment and analyze DNA in it, so you can tell what the organisms are living around. So it has a great potential to be used to detect different species and also to describe the whole biodiversity. So I think that making a gallery of different projects is very nice idea. It would be very interesting to see. And also I like the sense of a community, that the researchers maybe then can keep in touch and ask each other questions. And I thought that since I'm working with marine organisms, still maybe it's not the most common area, environmental DNA and marine organism. So maybe it's good to have one example of the project like this when we use a molecular tool in the sea and biodiversity monitoring.